All right. Well, thanks for coming, everybody. Welcome to Wave Club, um, organized by Josh Jacobs, me, Bart Ehrmantraut. That is, I'm Bart Ehrmantraut, Umath Mohan, and Irfan Zabeh. You can see their pictures there. Um, Today, we're happy to host Michael Breakspear from the University of Newcastle, and he's going to talk to us about the hippocampus as the brain's wave machine. Um, Basically, if you are interested, you can take a shot. We have a YouTube channel and we're on Blue Sky. I don't think we're any longer on X-Itter. Is that correct?
We are, but we're trying to shift to blue sky.
All right. And the logistics of this are that it's a 40-minute presentation, allows 20 minutes for questions, and you can use the Q&A features to type in your question, and then we'll bring you out to the stage at the end if you want. Each month has a different speaker, and self-nominations are very welcome. So today's speaker is Professor Michael Breakspear, who's a computational neuroscientist and practicing psychiatrist, leading the systems neuroscience group at the University of Newcastle and the HMRI. His research focuses on modeling large-scale brain dynamics and applying translational neuroimaging to understand schizophrenia, mood disorders, dementia, and healthy aging. He developed the Brain Dynamics Toolkit and his advanced methods to detect nonlinear brain activity from empirical data. He holds degrees in medicine, mathematics, philosophy, and a PhD in physics from the University of Sydney. He's held positions at University of New South Wales, QIMR, Berghofer, and now Newcastle, where he integrates basic science with clinical insight. In parallel, he provides psychiatric care, especially in recovery-oriented treatment and Indigenous health. His work has been recognized by major fellowships and national awards in neuroscience and psychiatry. So, without further ado, I'm going to let him um, get to it.
Let me just share my screen and then I can ready to go. It suddenly disappeared, of course. Um,
Just worked.
Sorry, everybody. It's just my talk. We just tried it and it disappeared. Oh, I know why. Okay. I can share my screen now. Um, Here we go. That should work. Okay, thank you very much. I'd like to thank Bard, Josh, Irfan, and Uma for inviting me to give this month's Wave Club. It's a very wonderful topic to speak to, something that I've been passionate about for many years, and I'm delighted to present some of this work because, of course, Bard has pioneered a lot of this work many years before the rest of us came. And I saw that there's a lot of people online who've worked in this space, but encourage you to go back and look at some of Bard's foundational work. The work that I'm presenting today is a lot of work that I've smashed into the next 40 minutes, and it's largely the work of my two postdocs here in Newcastle, Richa and Anna, and I hope they're both online and can ask some of the questions. So the outline of the talk is I'll talk about the functional embedding of the hippocampus and its various roles to begin with. Then I'll talk about the two models. This will be the main part of the talk of cortico-hippocampal waves, and then we'll have some time for discussion. Now, while I'm doing this, I need to minimize the Zoom window because I can't see what I'm actually talking about. how do I do that? That's just, okay, that might be better.
No, we are just seeing a small window. Oh,
Now you can see it again?
yep, perfect.
Okay, good. Okay, so I'm not going to labor on points that are almost self-evident to everybody in the meeting today, but we know that the hippocampus is involved in numerous cognitive functions, many of the core functions of the human brain. It's not doing these functions independently, but it somehow interacts with key cortical networks. And it's this interaction between the hippocampus and the cortex that really allows the execution of all these cognitive functions. Now, I'm going to give an example of a cortico-hippocampal this cortico-hippocampal embedding in an fMRI task, and I'm going to walk through it very quickly. But in this task, we asked people, first, before they went into the scanner, they watched 30 seconds of a newsreader introducing some newsworthy item. They did this 9 times, and then they went into the scanner and they watched the second half of these news clips. And this was the, what happened, that sort of reporter in the field half. And they watched another 10 that they hadn't seen the introduction to. But the details aren't really important because both during resting state and during the viewing of these news clips, every single voxel in the hippocampus, which is shown in the bottom right here, was functionally correlated with one or more cortical voxels. And every single cortical voxel, likewise, was functionally correlated with one or more hippocampal voxels. So during this task, the entirety of the cortex and the entirety of the hippocampus were covaried. And this persisted, by the way, after regressing out the actual task or the stimuli itself. So how is this correlation between the hippocampus and the cortex expressed? We did an algorithm called functional embedding, in which we looked at the principal functional gradients using a PCA-like decomposition of the hippocampus and the cortex. So we jointly embedded the cortex and the hippocampus, and we did a PCA, and we looked at the principal components of this cortico-hippocampal embedding. And what we got, which is what many people got, is that have seen is that there's this principal gradient that runs along the hippocampus, from the head of the hippocampus, which is shown in yellow, down to the tail of the hippocampus, which is shown in darker blue here. And this maps onto a similar gradient in the cortex that runs from what you might call the default mode, these higher order executive functional areas like the anterior pole of the temporal cortex, which you can see quite strongly, the angular gyrus, the precuneus, and the anterior cingulate cortex. So as you go from yellow to blue on the hippocampus, likewise you go from yellow to blue on the cortex. Or you can look at it in the other direction as you go from the tail. The tail is mapped onto sensory motor cortex. As you progress up to the head, you get into increasingly more heteromodal regions of the cortex. And this pattern on the cortex reflects or mirrors what people have published now for a decade following Daniel Margulies, that there's this principal gradient of functional connectivity within the cortex, at one end of which is the default mode and other higher order heteromodal areas, and at the other end is sensory cortex like V1, A1, S1 shown in dark blue. So there's this functional organization of the cortex, and the hippocampus is embedded in this overarching functional principle of how the cortex is integrated. And this isn't the first time people have mapped these too, but this is what we saw when people were just doing an ordinary everyday task, which is watching a news clip. Behavioral profiling of the hippocampus suggests that this gradient along the hippocampus from the head to the tail maps onto a self to world gradient. So the head of the hippocampus, which maps to heteromotor cortex, is sort of the egocentric component. And as we go down to the tail, we get into increasingly allocentric or world-oriented. I mean, it's a very general cognitive principle. I don't think it can be taken too literally, but you could look in the other direction. If you're on the tail of the hippocampus, you're mapped to sensory cortex that's oriented to the natural environmental milieu. As you progress up to the head of the hippocampus, the functional embedding on the cortex goes towards increasingly complex heteromodal regions of cortex. Okay, now that's a very quick and broad overview of the functional embedding of the hippocampus. What about the temporal dynamics of the hippocampus? Well, also for some decades now, it's known that on faster timescales that you won't see in fMRI, but you will see in intracranial EEG, the function of the hippocampus seems to be closely aligned to these 3 Hertz theta oscillation, 3 to 8 Hertz. So theta oscillations in the hippocampus are sort of an endogenous rhythm of the hippocampus, which is closely aligned to its role, for example, in encoding and retrieval of declarative memories. And there seems to be possibly 2 distinct functionally segregated theta oscillations in the hippocampus, a slow theta, sort of 2 to 3 Hertz, and a faster theta, 6 to 10 Hertz. I mean, I don't take these frequency labels too literally, and I think you can get a little bit over involved in naming them, but nonetheless, oscillations in these different frequency packets seem to be involved in slightly different components of the hippocampus function. And as I've shown before, different regions of the hippocampus map onto different regions of the cortex, and likewise, when you're looking at theta oscillations, phase locking of the hippocampus is functionally embedded into the theta oscillations in the cortex. So this mirrors what I've just discussed, which is theta oscillations in the tail of the hippocampus are phase locked to sort of sensory regions of the cortex, and theta oscillations at the head of the hippocampus are phase locked to prefrontal theta oscillations. Now, the theta oscillations don't occur sort of randomly when you're looking at space or as a standing wave, but seem to be organized in traveling waves that proceed up and down the hippocampus. So there's numerous papers now on traveling theta waves in the hippocampus. I've highlighted a few very nicely. Using intracranial EEG data in humans, people have been able to broadly estimate the wave speed of these theta oscillations and the wave length, but estimates, as you can see in these 2 studies, vary quite considerably. So we certainly haven't pinned it down, but I'll come back to that. And the directions of these theta oscillations, theta waves, reverse from head to tail to head, A to P to P to A. And moreover, that the direction of these waves appears to be important to the type of human memory processing that's being undertaken. So successful encoding is associated with posterior to anterior, and successful retrieval anterior to posterior. So now we're seeing And this is a paper by Uma, Bard, and Josh. So this is beautiful work that we're now seeing a lot of rich literature on the functional organization and the temporal organization of hippocampal theta oscillations and their waves. Now, my group and many others have spent some time really modeling the dynamics of the whole brain, and I'm going to quickly walk through this. And in the second half of my talk, I'll bring this to bear on how we're looking at these hippocampal waves that I've quickly walked through. So you can think of the brain on the left. The brain to T is some sort of dynamical system defined on these large-scale hippocampal and cortical oscillations and waves. So what are the underlying mathematical and physical principles of extracting out dynamic variables at the sort of meso and macroscopic scales that are relevant to the data that I've just been shown? The data I've shown is intracranial EEG and fMRI data. These are meso and macroscopic variables. They're not recordings of single cells or even small scale circuits. So we need to go back to physics and mathematics to understand, is there a way of modeling the large scale oscillations that we see in our data? So basically, I've cartoonized some histological representation of a cortical column. We can extract out some of the details and do a multi-compartment model where each single neuron is composed of many compartments using things like the cable equation, Hodgkin-Huxley-like dynamics. This is a sort of blue brain approach. We can extract out to the next level up and just have a single set of equations for each neuron, and then we've got spiking neurons and their circuits. The area that I've been working in is to then group together populations of these different neurons and set up some so-called neural mass or mean field equations at the mesoscale or even up to the centimeter scale of the macro scale. So this notion of neural mass modeling was pioneered by people like Walter Freeman, Paul Nunez, probably Bard, and then I'll walk through some of the work on neural field equations. But it's definitely not recent work. It's work that goes back 50 years, but it's become increasingly the focus of computational neuroscience. So I'm going to first do this cortico-hippocampal modeling using a neural mass model, and that'll mainly be the work of Anna Bieler, whose image I showed before. So before we show the cortico-hippocampal work, how do we do this mean field approach? So we, look at individual neurophysiology, which has been closely curated over many, many decades. But we consider at some level there's just many thousands of these in a region of interest. And kind of, but using more sophisticated maths than this, we use something like the central limit theorem to say, yeah, these individual neurons have complex nonlinear properties, but they're acting in a sort of ensemble, a little bit like a balloon of gas. We're not going to worry about the individual molecules in the balloon. We're going to look at a mean field reduction and get macroscopic variables like pressure and temperature. We're not throwing out all the individual neurophysiology. I'll show some work that I did with Max Shine's group that just basically demonstrates that if you tweak the physiology on the left, then these map into parametric manipulations of the macroscopic equations. So you might be tweaking the EPSP or the IPSP, its peak, its time scale, etc., etc. So we're certainly not throwing out the microscopic detail. Now, a simple model that I've used many years ago is a three-dimensional model with pyramidal cells, inhibitory potentials, and potassium channel conductance. This is based a sort of neural mass Morris-Lacar model. The details aren't really important, but if we manipulate the parameter, this neural mass model sort of displays what you can see on the left here. It's just sort of fast theta oscillations on top of which ride gamma oscillations. So this is a sort of theta gamma response. If we change the parameters into a different part of parameter space, this quasi-periodic attractor disappears and you get some sort of chaotic attractor. So we're going to take this chaotic attractor and put it in different regions of the cortex, and then we're going to look at the underlying structural connections between these regions and go from an individual autonomous system into an ensemble of coupled systems, which you can see on the left with a coupling function G. How do we inform this coupling function G that tells us how regions are coupled together? Well, in the last 15 years, we've seen dramatic advances in whole brain tractography, or we can go back to marmoset or cat or mouse, and we can inform it from detailed tracing studies. But most of the work we do just uses macroscopic tractography. So we have our autonomous chaotic attractor. We put them all over the cortex, and then we couple them together through an estimate of the human connectome, and we see what evolves. These are in the olden days where we used MATLAB, and this was a paper I published with James Roberts about 6 years ago, and we observed in this whole brain nonlinear model rotating waves, breathers, and traveling waves. Now, I'm not going to go into any further detail here other than to say these are not attractor states, but these are metastable dynamics such that if you watch these patterns for long enough, a rotating wave will turn into a breather, and a breather will turn into a traveling wave, and you get these complex metastable cycles. So if you're interested in that, James Roberts, Nature Communications. Now, whole brain waves have been observed in many different recordings, different species, different states of awareness. I think I saw Lyle Mueller online before, and so they've documented traveling waves in macaque. Why do we get these waves? Well, I put in the complex human connectome, but if we collapse that whole brain connectome into a function of edge weight or log of edge weight versus distance, you get an approximately exponential distance rule. Sorry, gone too fast. And this sort of frozen spatial noise pushes excitation in one particular node through its neighbors to more distant sites, and eventually you get these propagating waves. And this is seen in whole brain electrophysiological data. This is some ECOG data from Lyle and Terry Sinowski in a human at sleep. And to first order, we see sort of the temporal dynamics and the spatial wavelength and propagation velocity that matches. So that was work that we did. And now in the last couple of years, we've started to put the hippocampus in. So this is Anna's work. We take a high resolution hippocampus mesh. We're going to put a neural mass population at each mesh point. We're using the 3-layer Janssen-Ritt model. It's quite an old model, but according to the parameters, it does fast or slow theta. And we then couple them together using this exponential distance rule. We're not using connectomic data because we're looking at a very fine-scaled structure here. So we're just using an exponential distance rule. So if every node has the same parameters, the hippocampus very quickly converges to a global information poor synchronous state. But if we put some of these functional gradients that I spoke about at the beginning of my talk that we see in data, and so we break the symmetry of the parameter space with a gradient, we'll see something more interesting. But before we move into that, we can take the Kuramoto phase from each mesh point, and we can come up with a Kuramoto order parameter that tells us how strongly phase locked the hippocampus is. And the example I'm showing is very close to fully phase locked. And that is a state of global synchrony. But now we've varied the input current, so there's a constant current P plus delta P, and as we make delta P vary along the long axis of the hippocampus and increase the asymmetry, we begin to see these hippocampal waves traversing from the tail to the head or from the head to the tail. And as we do that, we see the Kuramoto order parameter begin to decrease because we're no longer getting global synchrony. So an imbalance in the external current following the principal gradient. We simulate this highly resolved cortical mesh. Then we take the phase at each mesh point and look at its surrounding neighbors, and we get local field vectors that tell us the direction and wave speed according to the length of the vector of these hippocampal waves. And using some simple maths, we can then start comparing the hippocampal waves to the empirical data that I showed before. Just before I do that, if we're in slow theta and the symmetry is from the head to the tail, the waves go from the head to the tail and likewise from the tail to the head. But for fast theta, the waves propagate in the opposite direction to the imbalance in the external current. So if the gradient's from the head to the tail, for fast theta, the waves go from the tail to the head. And maybe Bard can explain, because I'm not quite sure why we get that. So here's our hippocampal waves, and do they match empirical data? So these are the two papers that we went and compared against. As I said, the velocity and wavelength are not particularly accurately estimated at the moment, but certainly within a large region of our parameter space, we get waves whose wavelength and velocity are consistent with empirical data. So the hippocampus is done, sort of, at least in this model. What about its functional embedding? So we're going to take this functional embedding that I showed in our fMRI data and use that to couple each region of the hippocampus or each mesh point to a cortical region or mesh point. So here's our hippocampal model. We use an exponential distance rule independently on the hippocampus and the cortex, and then we couple them together using this functional embedding. In fact, when we go to these high-dimensional meshes, it's dense on the hippocampus, but it's sparse on the cortex because the cortex has got many, many more mesh points. So in the absence of coupling, here's our hippocampus and here's our cortex. We get complex structures on each. And now in a minute, Anna turned on the coupling, which you'll see in the arrow. And as soon as that coupling is enacted, you see a fundamental reorganization in the cortical waves. So this is the hippocampus. It's anterior to posterior hippocampal waves, and it's reorganizing cortical waves quite obviously and profoundly. So that's what I've just said. How do we study this across parameter space? Well, we can extract the Kuramoto order parameter from the cortex as well. And you can see how this captures what's happening in the cortex. starting from random conditions, we get low global coherence. Without the hippocampus, you get high global coherence, and then the coupling from the hippocampus starts, and it reorganizes into these waves of cortical waves. So what's happening with these cortical waves? I'm really running through a hell of a lot of work here with my apologies. We can decompose the flow of any surface using the Helmholtz-Hodge decomposition. This is actually a paper from a friend of mine, Petra Ritter's group, showing how you can get the flow on the left and the curl on the right. And so we can get the direction of the waves and the rotation of the waves. So Anna did this. We extracted the flow and used singular value decomposition to get snapshots of what these flow vectors look like. And you can see on the left, these waves are coming out of the front of the brain. Depends on the positivity or the sign of the eigenvector, and traversing to the back of the brain. And then they're sort of got a inferior frontal gyrus sink. And if you look at the time series of these three principal projections, you see a metastable fingerprint. So that's not a stable wave pattern, but there's different wave patterns coming in and out of appearance. This happens for slow theta. It also happens for fast theta when the waves are going from the head to the tail of the hippocampus. Try as we did, and this was Anna's work, we turned on posterior to anterior hippocampal waves, and we have not yet seen parameter variation in which it does anything to the cortex. So with the anterior to posterior waves, we get reorganization of cortical waves, but with posterior to anterior hippocampal waves, we get global cortical coherence with no interesting dynamics. So that's very interesting, and if time permits, we can come back to that. Okay, so that was the neural mass model, and now we're going to talk about neural field modeling. I know I'm going very quickly, but there's a lot of pretty videos, so hopefully you can follow those. So 2 or 3 years ago, James Pang and Alex Finito published this beautiful paper in Nature. Many of you might have seen, taking the cortical mesh, and instead of looking at activation regions using a general linear model, following work of Peter Robinson, decomposing this into cortical eigenmodes. Now, cortical eigenmodes are like Fourier modes or spherical harmonics that are adapted to the folding and geometry of the cortex. So there's a whole background literature here, but I hope you can see the picture that the spherical harmonics or cortical geometric eigenmodes are like a set of spatial basis functions for the cortical mesh. What James and Alex and Kevin Aquino and others, including Peter Robinson and myself, did is to take the Human Connectome project and take hundreds of subjects doing 7 different tasks: social, motor, gambling, working memory, language, emotion, and relational, and show that these geometric eigenmodes very quickly capture the variance and the different variants of these functional fMRI task-related maps. So you see by 100 modes, approximately 80% accuracy of these 32,000 mesh cortical maps. So that's 32,000 degrees of freedom captured in 100 eigenmodes. And these eigenmodes have very long spatial frequencies. So a consequence of this paper was to show that most of the energy in fMRI data, and by inference the human cortex, is not at fine scale, but at these very long spatial wavelengths. Now, the cortical eigenmodes don't come from nowhere. This is a damped wave equation, and this is now speaking to some work of Peter Robertson published in Neuroimage in 2016, showing that this damped wave equation can be decomposed into the spatial eigenmodes and a temporal dynamics. So this is a heuristic to decompose into time dynamics and spatial eigenmodes. So each of these eigenmodes at the top left have an underlying temporal dynamics, and that is what gives rise to these large-scale cortical maps according to neural field theory. So to show how the set of equations are closed, we've got propagating waves of activity across the cortex following a damped wave equation. This is a field phi. If we now move to some cortical area, this is incoming activity, principally into the superficial layers of cortex. That is filtered through the synapse and dendritic component. We're getting our EPSPs and IPSPs here, propagates down to the cell soma, activates some firing function, a sigmoid firing function, that then gets turned back into the field and propagates across the cortex. So this neural field model was published by Vic Yerser and Herman Harkin, and almost at the same time by Peter Robinson, who additionally added a time-delayed thalamic loop. So that's a whole brain corticothalamic neural field model. Here it is on the left. When I was a postdoc under Peter's tutelation, Peter told me, advised me to look at the temporal dynamics. So we've got something of the global mode that looks like a damped harmonic oscillator. And because of the corticothalamic loop, we get a resonant frequency at 10 Hertz. And if we go across a nonlinear bifurcation, we can capture grand mal seizures of the human cortex, which I've shown on the right here. So this was work 20 years ago, and there's been a lot of other work on the corticothalamic neural field model. But more recently, with the use of cortical eigenmodes, geometric eigenmodes, we can start to look at the spatio-temporal dynamics through the lens of neural field theory. So we integrate, now I'm going to show the work of Richer neural field equation using geometric eigenmodes. So each one of these modes is integrated, and then we pass it through the synaptic response function and the nonlinear gain from activation function, and that's the integration scheme. So this is the full brain corticothalamic model that Richard's coded up and mathematically analyzed. And so now we're looking at alpha rhythms, alpha oscillations expressed across the cortex. So that's the corticothalamic neural field model. We can do a geometric decomposition of the hippocampus. Jordan de Cracker published just a few years ago a high-resolution hippocampal sheet. So people in my group and others can do a geometric eigenmode expansion of the hippocampus, also put in a thalamic or entorhinal loop. And now we've got hippocampal neural field model. And it has a theta resonance, and that just arises through different gain parameters in this hippocampal thalamic loop. So now we've got a corticothalamic loop and a hippocampal thalamic loop. How do we couple them together? Their spatial geometries are extremely different. So Richer spent a lot of time mapping each onto a two-dimensional sheet, and then we can define a topographic coupling between the cortical neural field and the hippocampus field. This is the integral form. Richard did this. It's quite beautiful work in my opinion. So you get a hippocampal field. Sorry, it's reversed to what's shown at the top. And there's a sort of induced field transport through this two-dimensional embedding onto the cortex. So now we've got our complete cortico-hippocampal model using neural field theory. Here's a simulation of what it looks like. I've got to say, this is at least 2 years of work shown in 30 seconds or so. So now we've got alpha-like oscillations on the cortex and theta-like oscillations on the hippocampus. You can see that there's actually low-frequency undulations in both. So this is a full 1 on F-like spectrum with these superimposed oscillations. So as we increase the cortico-hippocampal coupling, the resonant frequencies in each begin to get more peaked and faster. You can see that here. Looking above, if the x-axis is the cortico-hippocampal coupling, you see this emergent phenomenon where the frequency within the cortex, which is in blue on the top, and within the hippocampus in red on the bottom, quickens because of this field-to-field coupling. Because of this conformal mapping, a region on the cortex will have its functional correlate on the hippocampus, and multiple regions on the hippocampus will have different functional embeddings on the cortex, which is exactly consistent with the work I showed right at the beginning of the work. So this conformal mapping from the hippocampus to the cortex is absolutely crucial to doing this because neural field theory needs to preserve the local neighborhood continuity. Here's the typical alpha rhythm on the left, which I showed before with the theta rhythm. If we increase this cortico-hippocampal coupling further, we go past another bifurcation and we get these high oscillation, nonlinear limit cycle-like activity on both cortex and hippocampus. And you can see that in the very peaked power spectrum, that it's not a resonance frequency anymore. And particularly now on the cortex, you can see the emergence of a rotating pathological wave corresponding to a wave along the hippocampus. And this goes back to work I showed that you do get these large-scale rotating waves in people with epilepsy. So to finally close the story, we had access to intracranial epilepsy data. Hippocampal seizures are very common. I believe one of the most epileptogenic regions of the brain. The work that I just walked through suggests that's because of the coupling between cortex and hippocampus. Here's an actual hippocampal seizure, which is propagated across to the cortex. And when you look at that seizure in the frequency domain, you see rapid frequency quickening followed by a frequency slowing. And we can emulate these dynamics and different seizure dynamics using our cortico-hippocampal model. And most importantly, this is due principally to an increase in the coupling between the hippocampus and the cortex, or an increase in the local nonlinear gain in the hippocampus. Where how much work that I've just run through. What does it all mean? I just want to close with this work I encountered in ticks very recently that suggests that when we perceive the world, we have this high dimensional representation on cortex, and to encode that as a working memory, we sort of need a dimension reduction, and this is exactly what is performed by by mapping a cortical representation onto the hippocampus. And literally, this is exactly what we're doing with our mathematical model, where we have highly resolved cortical meshes mapped onto a much low dimensional hippocampal mesh with much simpler dynamics on the hippocampus and vice versa. And then if you want to retrieve a memory, you take this low-dimensional hippocampal representation and you literally blow it up onto the cortex, as I showed with AP waves, reorganizing cortical waves. So I think this, if you don't remember anything else except for the pretty movies, this is the underlying functional embedding of complex metastable cortical waves, which much relatively simpler, lower dimensional hippocampal waves, and the wave-wave coupling between the two could possibly enact all these complex cognitive functions of the hippocampus. I'm not going to go through that because I've run out of time, but I'd like to finish by thanking Richa and Anna for all the beautiful work. There we are outside our imaging centre, and there's the rest of my group. Others who've worked on this include Saurabh, Nikitas, who's done a lot of the eigenmode decomposition, in Monash, of course, James and Alex. In Sydney, a whole bunch of colleagues, Mac, Eli, Brandon, Kevin, Ben, and Peter, and Jordan de Cracker for the highly resolved hippocampal mesh. So thank you very much.
Thank you, Michael. That was awesome. We have a bunch of questions coming in the chat. I'm just gonna moderate and have people come up and ask you directly. So first we'll take this question from Andrew Papale.
Hi, great talk.
Thank you.
I was wondering if any of the eigenmodes that you found were related to resting state functional networks.
Yeah, so James' paper did show that the functional connectivity of resting state fMRI data can also be very parsimoniously represented by geometric eigenmodes. And we're doing some work on cortico-hippocampal functional connectivity. So if you think in functional connectivity space, you've got this surface and you've got correlated activity with sort of edges between different vertices enacting that network. I personally worked in this space for at least 10 years. If you think of a geometric eigenmode, you don't have any magic lines or flight paths between the vertices. You actually have a single eigenmode and maybe 3 or 4 eigenmodes, and they're being expressed. So it's a purely cortical function. And the expression of those eigenmodes then, because they've got long spatial wavelengths, yields the functional connectivity networks that you see. Because if you've got the node of an eigenmode doing this, then the two nodes will be highly correlated, even though they could be at other ends of the cortex. Cortical eigenmodes and neural field theory does arise from white matter connectivity, this exponential distance rule, both within the cortex and through long-range white matter connections. But it's a complementary way of looking at it through geometric eigenmodes. But yes, resting state activity can be expressed in geometric eigenmodes. And exciting work that Nikitas and I are doing with Josh Siegel and others is on cortico-hippocampal functional connectivity, and hoping that we'll get that into the public space within the next few months.
Thank you.
Uma, you're on mute.
Sorry about that. Bard actually threw a question in here. Bard, since you're up, do you want to go ahead and ask?
Sure, I just wanted to know if you had distance-dependent delays at all in this.
Yeah, so the work I showed with corticothalamic, as the whole brain was just a single time delay. We have used distance-dependent time delays but not systematically. This is more the work of Vic Yersa and Gusteco doing time delayed. It's a complex issue though. When you're looking at long-range connections, many of the long-range connections in the cortex have a lot more white matter myelin, so they've got faster conduction, whereas shorter range are often unmyelinated, particularly in the cortex, within the cortical sheet itself. So you've got slower propagation sheets. But yes, you can throw in time, distance-dependent time delays. Neural field theory accommodates the time delays within the propagation, as I'm sure you know, Bard. So you've got implicitly distance-dependent time delays, and that's captured by the geometric eigenmodes.
Next, we'll take a question from Hyunho An. Do you want to come up and ask? Go ahead, whenever you're ready.
Thank you for your talk, Michael. Could you walk me through how your neural mass model handles the physical separation between the cortical nodes or hippocampal nodes? I mean, do you compute geodesic distance on the folded surface and use them to shape coupling strengths or propagation delays?
So for the neural mass work, as I showed, we took our fMRI data of people doing a task in the scanner, and we used that to inform which regions of the hippocampus were coupled to which regions of the cortex. And then we had a single parameter that increased or decreased the global coupling between hippocampus and cortex. And in this case, it was a one-directional so-called master-slave relationship. You'll have to ask Anna about the treatment of time delays. It's not a closed system, so it kind of doesn't matter because the time delays are only important if there's a reciprocal connection. But we wouldn't be using geodesic distance-informed time delays comparable to what Bart asked at this stage. As I've said, then you need a whole lot of data to constrain that. We could just look at the distance which we have, geodesic distance. We've looked at streamline distance, but there is not really, and I could offend people by saying this, the hippocampus is a fairly small structure, and if you're looking at diffusion MRI and try to get a connectome, you're not going to get anything like the spatial resolution of the model that I showed. We're talking about a few thousand mesh points, and that's not going to be resolvable with diffusion MRI. But therefore, I think you would just look at the Euclidean distance between cortex and hippocampus if you wanted to look at varying your time delays.
Thank you very much.
Thank you. There's another distance related question right after that. So it's a question from Harsh. Good.
Hey, I think you've partially answered this in your other answers, so I will modify it slightly. Is using exponential distance rule a simplification to make the model work because of the complexities of incorporating time delays along myelinated long white matter tracks? Or is there some advantage to using exponential distance rule as opposed to structural or functional connectivity?
Yeah, so this is a great question, and all I can quickly do is perhaps go back. I'm going to treat this question properly by bringing up a paper. I'm going to actually show what we did a few years ago. go to Zoom. This is a paper published with James Roberts a couple of years ago. So this is streamlined informed connectivity. This is weight versus fiber length, log of weight versus Euclidean distance, and log of weight versus fiber length. So you can see structural connectivity sits on top of an exponential distance rule. It's like a noisy perturbation of an exponential distance rule. We did quite a lot of other work on this hash, looking how much of the complex connectome can be captured by this exponential distance rule, put into a physically embedded cortex. And for first pass, quite a lot of it is. And the second pass, no, it doesn't do a great job. You know, the rich club, the highly connected nodes all just moved into the geometric center of the brain because they're closer to everything. Whereas we know from structural connectivity, there's clusters in precuneus, there's clusters in other cortical regions. But this is like a first order perturbation of the exponential distance rule. So you get a long way just with the exponential distance rule. If you want to look at the first order perturbation, you need to start putting back in structural connectivity. And we're doing that, obviously. And you do get interesting perturbations in the dynamics when you start putting in these long range connections that are perturbed off the exponential distance rule. So that's a really interesting area. Obviously, they're in the cortex. They come out of these large pyramidal cells in deep cortical layers. They, for many reasons, use more energy. So possibly the brain is humming away on the exponential distance rule. If you want to change from one state to another, you enact these long-range perturbations and you destabilize the cortical eigenmodes. Then you can stop doing what you're doing and transition to a new state. But to first order, yeah, in fact, for the neural field theory, it's a Bessel function of the second order, which is very, very close to an exponential distance rule.
Great. Thank you so much.
Thank you. We just had a question. Yes. Robert, you're up. You go for it.
Hey, Michael. It's related to also like the follow-up question by Rainer. So in your last slide of like what this all means, is there a way to test that by using TMS to do some downregulation someplace on the cortical surface?
Yeah, look, great question, Robert. And there are there's a few people, including, you might have seen a paper from John Griffiths and Davide Monti doing TMS with neural mass models.
Okay.
Also the work of Luca Cocchi and colleagues, which Andrew Zaleski and myself have looked at TMS in perturbations, and also work of TMS perturbations in neural field equations. That's something coming that Luca's working on. What I'm interested in are other neuromodulatory perturbations, and hopefully in the next few months you'll see a preprint on some psychedelic perturbations of cortico-hippocampal coupling, and then I think our cortico-hippocampal approach works beautifully well. But I can't speak of that at the moment because all we've got is the work's been done and the figures are there and the words haven't been finished putting around it. But yeah, perturbations of TMS, DBS, or neuropharmacology are really interesting ways of analyzing. The other approach has been sleep and epilepsy, which are state transitions. And I saw Lyle put up a point here that their data was, yes, it was patients with epilepsy, but they were sleeping. But Lyle, there is other work looking at rotating waves in epileptic seizures as well. So all these perturbations are important. And it's just a rich area to be working in, really. I encourage people to get stuck in because there's a lot that can be done here.
Awesome. Thank you.
Well, thanks for coming up. Do you want to ask your question while you're here?
Sure, yeah. I just wanted to note that the that in the sleep spindles, those were physiologically normal, and that's not to say that there could not be rotating waves and the epileptiform activity. But I think it's important to distinguish between those two types, right? Because we know that they have very different physiological features. Um,
Correct, yeah. So I've patched those together a bit messily. There is in vitro recordings of epileptic-like activity with rotating waves in pathological oscillations, but this is not in vivo.
that's correct. Yeah, that's and Jen Young Wu's group did a lot of the early research on that. But that's at a very different scale, right? Because that's not only that's slices and only a few millimeters, right? So it's really a fundamentally different spatial scale there.
Yeah. In our neural field work in the physiological range, we do get glimpses of rotating and traveling waves. I mean, it's a damp wave equation. It's just a lot of the eigenmodes are superimposed, so you don't see them so so clearly.
I wanted to ask a question, if I can ask an additional follow-up question, about the fixed time delays as well, because so in the Roberts paper, the waves appear to go across the cortex in roughly 6 or 10 milliseconds or so. And I think the speeds there are on a median of, let's see, 26 meters per second, which is really relatively fast. And I think we've seen some differences when we're using distance-dependent time delays going along Bard's point, where we really see the waves going more in the 2 to 5 meters per second range. So I wanted to ask if you could clarify that point, how fast are the waves going in these models, and what is the role of the fixed versus distance-dependent time delays?
Yeah, look, if you look at the neural mass work that you're talking about, the Roberts paper, we did do some mapping of the state transitions with MEG data from Mark Woolrich from a hidden Markov model. But we did note in that paper that the transmission velocities are very quickly. Yeah, you can slow down the waves with transmission delays. You can destabilize the waves with transmission delays. And Carlo Lang has shown that when you go from an exponential distance rule and you add this spatial noise, you also slow down wave propagation in general. Now, there probably are more than one set of waves in the cortex, right? There's calcium waves, there's waves carried on slow theta, there's possibly gamma activity being carried in very fast waves. A lot of our waves in that paper were high frequency waves, high temporal frequency waves. What's probably more powerful about the neural field theory is that the damped wave equation can be parameterized with the propagation's velocity. I raced through that, obviously, but there's a parameter in there that is the wave speed. And you can take empirical recordings and build that into the neural field equation by putting it into the damped wave equation. And that's why in my own work, I mean, I worked on neural mass and connectomes for many years. Neural field equation is probably an area that I'm increasingly going back to because of its physical and physiological grounding. I mean, that's why it's so much harder to couple the hippocampus to the cortex with neural field equation, because the neighborhoods, the hippocampal mesh, have to have continuous neighborhoods on the cortex. You don't have to worry about that with a neural mass model. And that's why Richard had to do all the conformal mapping. The wave velocities that you've just spoken about have to be informed by actual wave velocities. You've got alpha, you've got gamma, you've got beta frequencies, but they're on a 1 on F like spectrum. They don't just jump out of nowhere. And you can only get that one, well, we get that 1 on F spectrum from the wave equation itself. So looking at physiological realism, I think neural field theory is probably the way to go. Looking at understanding underlying principles and wave propagation and all of that, neural mass models sort of easier to get your teeth into. But you'll get mismatches with data, which is, I guess, what you're pointing out, Lyle.
To some degree, and I'm happy to chat about it more offline.
Yeah.
Thanks.
Thank you, Lyle. There are a few more questions. Michael, I just want to make sure, are you okay on time, or do you want to wrap up and take them offline?
I've got till 9:30. I apologize. Someone's using a concrete saw in the house next door.
Okay. Then maybe we'll do a couple more and then wrap up.
Sure.
Okay. There's a great question from Elliot Smith. Do you want to go ahead and ask? Oh, I hope he's still here. Oh, he might have left, actually.
So to look at the chat, yes, the waves are on mesoscopic activity, not single neuron activity. They're LFPs and they're macroscopic field potentials, depending. Neural mass gives you more LFP, neural field gives you EEG, MEG scale. Corticothalamic coupling is crucial, but a point that I glossed over is there's no field within the thalamus. There's no field propagation. Each thalamic point has its cortical point, its cortical neighbor, but there's no within cortex damped wave equation. There's a damped wave equation on the cortex and the hippocampus, but not within the thalamus. And that's an important point because the thalamus can quickly get very rough spatially, and that can start adding a lot of noise back into the cortex and hippocampus. I mean, you could take the thalamus if you had a surface rendition, do a geometric eigenmode, and do a thalamic neural field model, but I haven't seen the purpose of doing that. So yeah, that's the answer to his two questions. Rainer Engelken, is he still here? Maybe he could.
Yeah, looks like he just asked a follow-up.
Hi.
Rainy, yeah, go ahead.
Yeah, thanks for the talk. Yeah, I was wondering whether you could make some concrete, experimentally testable predictions of your theoretical work.
Yeah, look, it's a really interesting question. I mean, we can go on and on parameter tuning each time we get different empirical data and match it to the empirical data. But that's not really testing, if you
Yeah, that would
like.
be a prediction. I was asking
Yeah.
for prediction.
Yeah. I think the neural field modeling is stronger in that the neural field model has been trained on alpha rhythm, has been trained on epileptic seizures, hasn't been trained on cortical-hippocampal activity. You will see in the preprint that there is a theoretical prediction with some nice algebra from Peter and Richer that says if you increase cortico-hippocampal coupling, you'll get a multiplication of the gain in each system and you'll get an epileptic seizure. And that's what we did. And that's how we get our cortico-hippocampal seizures. Yeah, I mean, we could then, I mean, it's physically constrained. So say we take our psilocybin data and we took empirical independent estimates of what psilocybin does on neural response functions, that would be a prediction about what we would see when we put those parameters into a cortico-hippocampal neural field model. It's getting closer to it's getting closer to prediction. Yeah, I think you could predict these sort of things. You can predict the effect of TMS. At the moment, I feel like we're still stuck in we're trying to convince people how powerful these theories are mode. But Peter Robinson, my former postdoc supervisor, is very strict that these are predictions, Michael. The neural field model is there. It's got physically constrained parameters, and it can predict this and it can predict that. Yeah, so that's kind of where we're at the moment.
So, if I could ask, you said one prediction would be if you increase the global coupling between hippocampus and cortex, then you get epilepsy. Is there a way of doing that, or how would you actually like test that prediction?
Well, I guess you go to people with hippocampal seizures and you find either abnormalities in the hippocampus or abnormalities in the structural connections or functional connections between cortex and hippocampus. I'm told this is why the hippocampus generates so many seizures, that you need relatively minor perturbations to the integrity of the hippocampus. And because of its relationship to the cortex, it'll start throwing off seizures. Whereas you can get a perturbation in the cortex and it may not cause a seizure. It might cause a local seizure, but it's not going to cause a global seizure. Um, uh, you could likewise take someone with epilepsy and maybe you can do DBS of the hippocampus. Or maybe you can take someone with dementia. We know people with Alzheimer's disease often get hippocampal seizures. Maybe we can disrupt that and we can preserve cognitive function. Um, it's the sort of work that we're going to be doing, I think, in the next 5 years. You could take a non-human rodent model and maybe do some more invasive perturbations. That's not in my work, but other people could do that.
That's
You're
awesome.
now talking about activity perturbation, but your predictions was about the coupling strength.
Yeah, I mean that's a strong prediction, and it's borne out in the numerical simulations that Richard has done that I showed. How do we take a brain and artificially increase the coupling between the cortex and the hippocampus? I mean, that's maybe what psilocybin does, but that's a whole brain effect. I mean, you see in those papers from Josh Siegel and others, massive increases in functional connectivity between cortex and hippocampus. But what causes that? If you want a generative model of that, you have to do neural field theory or neural mass models, in my opinion.
Very cool.
Thank you.
We'll just take one last question and then we'll wrap up. There's a question from Alessandra Yu about coupling across frequencies.
Yeah, look, that's a beautiful question, Alessandra, and I'm pleased to say that Richa, who's online, has done some beautiful work on this, which I can hopefully briefly show. Share. I know Richard's online, but look at the beautiful work of this is what's going to be in the paper. Two answers to that are yes, even in the physiological range, when you increase cortex and hippocampus, this is what I showed before, you get an increase in the rhythms, but you get the emergence of this new frequency in the cortex that seems to arise here. And so these are sort of off-frequency effects. So cross-frequency coupling, which is what you've alluded to. You don't get in our work, well, you do get gamma theta coupling, but that's a different story. If you look in the physiological range, the mode coupling, which I show on the right, which Richard shows here for the cortex and the hippocampus, is very weak. When you get near or above the bifurcation, you get very strong mode coupling, which is not temporal frequencies that you're alluding to, but spatial frequencies. So you get high spatial frequencies coupled with low spatial frequencies, and you also get these temporal frequencies coupling. So you get very rich mode interactions in these cortico-hippocampal neural field. It's a rich tapestry that we're barely scratching on. And I have to tell Richard, no more. We're going to publish this work. We've been working on it for a couple of years, and there's oodles of work in there. But you'll see that when it comes out.
Thank you. It's really interesting.
Very cool. Excited to see that out soon. With that, we'll wrap up. Thank you, Michael, so much. This was a great talk. Anything else from you guys?
Thank you, Michael. It was great. A lot of work.
Yeah,
Yeah.
we don't travel as much as we used to, so the talks tend to get more dense again. But I'd like to thank Josh and Bard and Uma and Irfan for inviting us, inviting me to present our work. And since I'm finishing, I'll just finish with this acknowledgement. I should have put at the beginning, but nonetheless, it's gone. Well, I'd like to acknowledge the Warramai and Awabakal people on whose land we're working on here in Newcastle. It's actually Awabakal and Warramai nations go back many thousands of years, and we've given these English place names, but they've got they belong to they're unceded lands of Indigenous people with names that actually link to the country itself.
Thank you for sharing that.
Yeah.
That's an important acknowledgement. All right. Thanks, everyone.
Thank you,
Okay.
Michael.
All right.
Thank you.
Great.